How's it going, everyone? Today we are going to talk a little bit about vectors in biotechnology, so let's get started. Okay, so vectors are pretty fun because they're very crucial in biological technologies, but the basic question that you have to answer is that let's say you have recombinant DNA, and that recombinant DNA can be used to clone genes, but you need to get it into a host cell. To get it into the cell, that recombinant DNA can either be integrated directly into the host chromosome, or it can enter the host cell with some kind of a carrier sequence, that's called a vector. So that's what basically a vector is. It's just this secondary integration of some kind of a DNA sequence, into a host cell. So in order for a vector to be of good function and good use, you need a couple of things to happen within your experimental procedures. The first one is the creation of that recombinant DNA of interest, so that particular strand of rDNA that you want to have expression of in some sort of way. And usually this includes some kind of a reporter gene. You obviously want something to show you that that gene is going to be active by having some kind of a gene near it potentially that can report and say, hey, this particular gene is actually being transcribed. So some kind of a reporter gene or selectable marker to help you out in the process of identification purposes. In addition, you should have some kind of a vector with proper insertion into the host cell, so potentially a unique restriction site. And finally, very important potentially, is this ability for autonomous replication in the host cell. Now, when it comes to choosing the host cell, that depends on what type of model you're working with, but the host cells can be plants, they can be cultured cells, bacteria, and maybe yeast as well. When it comes to vectors in total, there are actually kind of different types. So we often talk about two main types of vectors in biotechnology. The first one is probably a very well-known one, and that is the plasmid. It's just a circular sequence of DNA. Some people call it a quote-unquote chromosome that basically replicates separate from the host's own genome. So it replicates separate from the actual bacterial or host DNA. So oftentimes when we talk about plasmids, we're going to be targeting bacteria such as E. coli rather than really eukaryotes. And this process of inserting a plasmid into a bacteria, for instance, is called transformation. There's another type of vector available, and that is the virus. So we have things such as bacteriophages or retroviruses. So this is a bacteriophage over here. And the virus can basically be used to attenuate some detrimental genes in the virus. So you can go ahead and take those particular that DNA sequence and can be altered. So that virus can be altered to attenuate some detrimental genes, And you can also alter it to insert some kind of recombinant DNA into a particular host cell. So if it's a bacterial phage, the host cell would be a bacteria, but you could obviously do it with eukaryotic and maybe mammalian cells as well. So the infected cells often will express some kind of a different phenotype for easy identification purposes, and target organisms include animal cells, but also technically bacteria as well in the case of bacteriophages. And this process of putting, for instance, some kind of a DNA or just a nucleic acid sequence using a viral vector, so using a virus to inject a nucleic acid sequence into a bacteria, that's called transduction. So just some vocabulary there. Now, when you take a look at the vector components themselves, for instance, with a plasmid, you need some things within that vector. And the first thing is this origin of replication, which is pretty important, I'd say. You also might want some reporter genes, as discussed, or basically genes whose expression is easily observed, so you know that the plasmid was taken up by the host. In many labs, we use a green fluorescent protein. And these selectable markers can also be used to select for certain bacteria. So you can also put in certain selectable markers like resistance to certain antibiotics, and so those cells that didn't pick up that particular plasmid will basically not survive if you grow it on a medium that has ampicillin or some kind of antibiotic on it. So if you look at this particular example here, there are two types of reporter genes, and these include your ampicillin resistance genes as well as your tetracycline resistant genes. So again, this allows you to know if the plasmid is basically properly taken up in the bacteria and if the plasmid is going to be recombinant. Oftentimes, in addition to all this, you also have a promoter before the gene of interest, and that's going to be pretty interesting and intriguing and important because you obviously need that promoter potentially to get the whole process of transcription started. There are a few ways you can actually get that DNA into a cell. One of the methods is actually with just transformation, where you're basically taking that recombinant DNA and inserting into a cell with a non-viral transfer. So you're not using a virus. So this is more of a plasmid type situation with transformation. The problem is that oftentimes only a few cells are actually transformed. So maybe only 1 in 10,000 cells are actually going to be successfully transformed, which is why you need a selectable marker to basically eliminate the cells that didn't get transformed. There are two main types of ways to basically insert DNA. The first one is just simply treat those cells with chemicals to make the membrane more permeable, and DNA just diffuses into the cell. The second method is with electroporation, where you're actually using electric shock to make temporary pores in the membrane. And then finally, last but not least, I just want to mention these two terms over here. We have transfection, which is basically insertion of DNA into a eukaryotic cell, which, for instance, an animal cell or maybe a plant cell. So this is often known as transfection, which is just another word or another term for it. And then finally, we have transduction, which is viral-mediated transfer of genetic info into a host cell. So you're basically using a virus to help mediate that genetic transfer from the virus into the host cell. So it can get a little bit more complicated with these terms, but just keep in mind that transformation, you're often using just plasmids, using those plasmids with the recombinant DNA and inserting it into the cell without some kind of a virus. Transfection, simply taking that DNA and then inserting it into a eukaryotic cell. And then transduction is you're using some kind of virus, right, to help mediate the process of genetic transfer into some kind of a host cell. So that's basically all I have in terms of vectors. I hope you found this mildly useful, and as always, best of luck studying.